So for the exam, in terms of replication, and all questions are welcome, not necessarily just about content of the exam, but we can talk about other things for the exam. For replication bubble, the important things to understand, yeah, there might be a labeling question. It's important to understand the process of DNA replication. So for example, it's important to know what the enzymes do that are involved in DNA replication. You should be able to do the polarity, labeling, location of primers. Things that you might want to remember are RNA primers are RNA. They'll have U's instead of T's. That's the main way to distinguish primers from synthesized DNA. Good thing to remember. Replication bubbles, forks go in both directions from origin of replication. So you start with an origin that's small, comprises maybe 20 or 30 nucleotides, and then grows as the replication forks expand in both directions. Pretty soon you get a really big replication bubble. And eventually that bubble will fuse with replication bubbles on one or both sides of it as chromosomes complete their replication. You want more clarification on that? Okay. Maybe we'll get to more replication questions as we proceed too. Okay, so what enzymes did we talk about? They're in the list of vocabulary for this chapter. Here's a replication bubble. Where are the enzymes and what do they do?
Is it helicase, the one that makes the bubble bigger?
Okay, so we've got helicase, which an ACE is an enzyme that catalyzes something. So in that case, it's the unwinding of the helix, helicase. So it's at both of the replication forks. And it's the enzyme that breaks the hydrogen bonds at the replication forks and helps the double helix become two single-stranded pieces of DNA. What enzyme is it that builds the RNA primer that base pairs with the single-stranded template?
Is it a primase?
Primase. Yep. What other enzymes are involved in replication of chromosomes? Gyrase, polymerase, ligase. So that's three more. I think that might be it. I might be missing one. So we'll go through those in turn. You know what it means to gyrate, right?
Okay.
You want me to gyrate? I would be happy to demonstrate, but you might not want that. Gyrase means to twist, to rotate. That's the enzyme that we did a little demo up here with a rubber band and a pair of scissors. That's the enzyme that works downstream of the replication fork that will clip one backbone, one strand of DNA to let the torsion unwind that causes when you open the replication fork. The rest of the DNA becomes supercoiled, and gyrase is used to relieve the tension so that the chromosome doesn't break under the tension of that twisting that happens in the double helix. Polymerase. So we're going to grab on to one end, the 3' end of the primer, and that's the enzyme that actually adds nucleotides onto the end of the primer. That's the synthesis of DNA. That's the creation of the new strand of DNA. What was the other one? Ligase. So that has to do with the lagging strands. So in this case, I've drawn polymerase on the top here, grabbing onto one end of the primer. So that, by definition, must be the 3' end of the primer, which means polymerase is moving that direction along the top strand, adding new nucleotides, which means there are going to be some primers here on the upper left side of that top strand that are going to be primers that prime the lagging strand synthesis. So each one of those short RNA primers will spawn a little bit of synthesis of DNA from a separate molecule of DNA polymerase. And the problem with the end of DNA replication is that when DNA polymerase adds that last nucleotide at the end there, that polymerase does not covalently attach that last nucleotide to the start of the RNA primer. So there's a physical gap in the nucleotide phosphodiester backbone there. That's what DNA ligase does. It's a separate enzyme that recognizes nicks, breaks in the phosphodiester backbone, the backbone of one strand, and catalyzes that reaction. It connects the phosphate and the hydroxyl back together. So the end of that nucleotide would have a 3' hydroxyl, and the start of the RNA primer will be a 5' phosphate. So this ligase catalyzes the chemical reaction that covalently bonds that hydroxyl and the phosphate together. So it makes a continuous backbone of that strand. And it does that every time there's a break in the backbone, which is where every lagging strand synthesis step has completed. There's always going to be a break there. Ligase has to come in and reconnect the backbone. Gyrase, polymerase, primase, helicase, ligase. Were there any others? I think that's it. Don't need to know topoisomerase. It is an enzyme that's involved, but we haven't talked about it.
Is topoisomerase different than gyrosin? Or is gyrase a type of topoisomerase?
A gyrase is a type of topoisomerase. So yes. How to clarify without making this more confusing for everybody? A gyrase is a type of topoisomerase. A topoisomerase is a class of enzymes of which we've seen one, gyrase. But we don't need to know the other members of the topoisomerase group.
Can we do like number 11 practice mock exam? It's like the gapped multiple sequence alignment.
Okay. Gapped multiple sequence alignment.